Donc, bonjour à tous et à toutes. Bienvenue à cette dernière conférence de la série de séminaires de l'ISFORT. Pour les gens du cours de Sujets de pointe, il nous reste encore un cours, mais c'est un cours où on lit un article, donc il n'y aura pas d'autres séminaires avec des invités. Aujourd'hui, on a le plaisir d'accueillir quelqu'un de loin, mais on l'accueille virtuellement à l'ISFORT. On accueille Frida Piper, qui est professeur à l'Université de Talca et aussi affiliée à l'Institut en écologie et en biodiversité. Donc aujourd'hui, Frida est une écophysiologiste, donc je vais lui laisser la parole pour écouter sa présentation. Puis vers à peu près 1 heure, il y aura une période de questions ouverte à tous. Puis pour les étudiants du cours, on va se retrouver à 2 heures pour faire la séance de discussion en ligne avec notre invité. So Frida, I'll let you get into your presentation. And like I said, I'll wave around 1 o'clock so we manage time properly.
Yeah, thank you. And I should say good morning, I think. So thank you for attending my presentation and for this invitation. Sorry. And yeah, I will present a very recent, still unpublished study, which is entitled "Do High Levels of Carbon Reserves Shape Local Adaptation to Drought in Temperate Trees?" This is a seminar for the Institute for Temperate Forest Science at the University du Québec in Ottawa, Canada. And it is at the same time part of a lecture for the PhD program in vegetal biology and biotechnology at the University of Talca, where I participate. As Audrey mentioned, I'm currently working at the Institute of Biology Science in the Universidad de Talca. I also belong to the Institute of Ecology and Biodiversity, which is a multidisciplinary institute with multiple locations, represented by a group of researchers from Chile. However, I'm living at this part of the country for only 1 year. So most of my previous work, including the work that I will present today, comes from my career in Patagonia, in southern Chile, where I spent living and working for more than 10 years. So let's introduce the topic asking what are carbon reserves. All of us know that trees get carbon through photosynthesis, and the destination of these photoassimilates can be an immediate consumption in growth, respiration, or defenses. Can be an immediate use, but not necessarily involving consumption, which is, for example, the role of sugars as metabolic intermediates or as osmotic compounds. And a third, much less studied destination of photoassimilates is the formation of carbon reserves, mainly represented in plants as starch and fructans. These 3 main sinks of photoassimilation can be played in the crown of the tree, in the trunk, and also in the roots. And most of the 3 sinks are exchangeable among them. So sugars can be mobilized from the carbon reserves and be used to growth, to respiration, to defenses, or as osmotic compounds or metabolic intermediates at any times. And of course, this can happen in all the organs of the trees. An important part of the carbon gain is lost as exportations, as a biomass loss, or as a root exudates. And we talk and we study about carbon because carbon is the main element compounding plants, especially trees. Store the formation of trees. This is forest, including tropical forest and temperate forest, store close to 90% of all the biomass sequestered as carbon on Earth. So it is very, very important to know to understand the dynamic of carbon when we think about the increasing CO2 atmospheric concentrations that we are facing in the most recent years when we compare to the 600,000 years ago, where the concentration was not as much as 300, but now we are reaching 400 right now already. And we look also on the destination, on the fate of carbon, because sorry, I will turn off my cell phone. Because at the current CO2 atmospheric concentrations, the rate of photosynthesis of most plants on Earth is not saturated. So there is a leeway. There is a leeway for increasing photosynthesis and potentially for increasing carbon sequestration through the formation of biomass. But a question before this is to know whether tree growth under natural conditions is carbon limited. Because we can expect increasing carbon sequestration in the future only if we know that growth is carbon limited under current conditions. And here is where I started to get interested in the use of carbon reserves as an indicator of the carbon supply status of trees, especially in terms of comparisons. We can do comparative analysis of carbon reserve concentrations in plant tissues under different conditions, for example, defoliated trees and non-defoliated trees, trees under drought and trees under control conditions, or trees under shade, or trees under good light availability. And so the expectation is always the same, is if trees are carbon limited under any of these stressful conditions, we could expect lower levels of carbon reserves under those stressful conditions than under the control conditions. So, and this is because the carbon reserves reflect a balance between the gain of carbon through photosynthesis and the use of carbon for the different functions that we saw in the previous slide. Nonetheless, a part of that carbon is sequestered, is considered sequestered, is not available for use, so it's really not available. We don't know the reasons of that, but the fact is that almost all studied trying to starve plants find that plants die having some level of carbon reserves. They hardly ever die with zero carbon reserves. So that part of the carbon reserves is considered sequestered. So we have that expanded carbon reserves concentrations will occur whenever the carbon influx from photosynthesis outbalances the net usage of carbon, for example, for growth, for respiration, for exudation, or even for the formation of reserves itself, which is something very, very novel and very controversial. So let's put it in a side for a while. And then we can have shrinking carbon reserves if the demand for carbon assimilates is higher than the current photosynthetic carbon activity. Therefore, we can expect that if the growth of trees is carbon limited under any condition that you won't consider, for example, dry conditions, recent drought events worldwide, well, if there is a carbon limitation on the growth of trees, we should expect a lower level of carbon reserves in those trees when compared to more humid sites or more humid years within the same site. When we talk about carbon reserves, we are referring to the mobile carbon pool. So this is to put it in contrast to structural carbohydrates, which are represented by cellulose, hemicellulose, lignin, which they are not useful to provide energy as the pool of mobile carbon is. And this pool is represented, is also named carbon reserves or carbon storage, and is mostly represented in plants, as I said before, by non-structural carbohydrates, which can be very often in the literature named as NSC or TNC, from total non-structural carbohydrates. And they are mostly composed by polysaccharides, so very big sugars, very big molecules of sugar, which are starch and fructans in some plants. Normally, starch is the most dominant in different plant families, but some families, like Compositae, prefers to store sugars in the form of fructans. And we also include in non-structural carbohydrates mono and D or oligosaccharides like glucose, fructose, and sucrose as the three most common. And then we have sugar alcohols, organic acids, phenolics, isoprene polymers, and terpenoids, which are also included in the family of mobile carbon pools. Generally much less represented when compared to non-structural carbohydrates. And we also know that lipids are a preferred form of carbon reserves in some plant taxa, particularly in conifers. So there is very, very well known nowadays that all conifers, and potentially more species that we still don't know, but from the current knowledge, it is clear that lipids are the preferred form of storage by conifers. So let's talk now that we know how they are more or less regulated, how are carbon reserves formed or conformed. So now we ask, why are they important for trees? So we know that they are used whenever the carbon balance is negative. Yeah, so seeing in those situations in nature when you have this negative carbon balance, yeah, I really like to show the picture of the left side, because it's in my opinion, an illustration on how carbon balance make prolonged life after life, or may mediate resurrection in plants, or may mediate this modular concept that we know from plants. Yeah, so plants resprout even when we think that an individual is completely dead. They can resprout, and the energy for that resprout comes from carbon reserves. So this is a picture of a fence made by stems of poplar in Patagonia. And you can see that in springtime, all these poles used for in the fence are becoming little or weird kind of trees. And this, of course, is important for trees to face and to overcome very, very ecological relevant filters like fires, or like herbivory, as we can see in the pictures of the right side, where I am showing this first in the here you can see this caterpillar, native caterpillar in the Andes, which is called Orniscodes amphimone from the Saturnidae family, which is responsible of this amazing forest defoliation you can see in the lower panel. Can you see the arrow of my mouse? Yes, okay, good.
Yes, we can.
Great. So this is this here you can see, of course, the deciduous forest in the Andes, which is dominated by only one deciduous species of Nothofagus, and it's autumn time, and this is because you see here the red crowns of the trees, which are still with leaves. And here we cannot see leaves because in the previous period, this is in summer and in late summer, the caterpillar eats the whole forest below this isotherm, probably due to temperature limitations to this insect. But what was really interesting to me from this system is to see how the trees were producing little small green leaves in the middle of the autumn. Yeah, so they were in a way adapted to this disturbance. They were producing these leaves, trying to get some photosynthesis because before the long winter arrives. So, and in another groups of studies, I was focused on the strategy of these species to manage the carbon pool in order to be adapted to this very catastrophic and recurrent defoliation, and eventually between this love relationship between the tree and the caterpillar, which is sustained in time, like a very good marriage. So they arrive to a very good business to do that. And it is a business mediated by carbon reserves. And because of these roles of carbon reserves, you probably can already imagine that both non-structural carbohydrates and sugars are a trait, a life history trait that increase the plant survival when they are under carbon stress. So this is a finding of a recent study that we published, where we show that in seedlings of very different plant families and functional types, like an exotic, deciduous, winter tree species, and evergreen native tree species, and also a native deciduous species, and then a conifer native species, all of them were able to increase their probability to survive to carbon stress, manipulated through shade and defoliation. According, they had greater contents of non-structural carbohydrates or soluble sugars. And a similar finding was a year before showed by a group of the United States with oaks, in mature trees of 2 oak species, where they found that below a critical threshold of total non-structural carbohydrates, the oaks died. Yeah, so very, very similar results in quite contrasting conditions. And more recently, it was demonstrated that the plant carbohydrate storage in plants is hereditated. Yeah, so it's a trait under heredity. And therefore is posited by this study as a major life history trait. I would, however, like to say that if you know the studies of Grime, yeah, was Grime, Philip Grime. He already mentioned this trait in the comparison of stress-tolerant species and competitors, yeah, where we would expect stress-tolerant species to allocate much more carbon to reserves in order to tolerate stress. But for some reason, during many years, the ecological consequences of carbon reserves was not very considered. In the same line of about the importance of carbon reserves, we know that globally plants in different biomes and including different functional types like conifers, evergreens, dry deciduous, winter deciduous, and herbaceous plants keep constant minimum non-structural carbohydrates levels over the year, keep the same level when it is expressed relative to the maximum. So when plants have higher maximum of non-structural carbohydrates, they have higher minimum, yeah? And saying the same thing, but in a different way, this is that the minimum non-structural carbohydrate concentrations as a percentage of the maximum non-structural carbohydrate concentrations is around 40–46% for most plant species, yeah, especially for total non-structural carbohydrates and for soluble sugars. Yeah, this is a meta-analysis study where I participated. So we analyzed more than 250 species all over the world, and including different organs. And one of the main findings was this that I am mentioning. So that most species had a minimum NSC and a minimum SS concentration between 40 and 50%, which strongly suggests that globally plants really need to account for carbon reserves to survive wherever they thrive. But let's then now move to the role of carbon reserves in relation to drought. So we know that they can work as an energy supply, for example, mediating drought avoidance when plants shed their leaves, or when they close their stomata for a long time. They have to get energy from somewhere, so they get the energy from the carbon reserves. And therefore, we can think that to account for carbon reserves should be advantageous to face drought. They also play a role in the tolerance of plants for under dehydration, scavenging, for example, the reactive oxygen species, or protecting the membranes of the cells. And they also play a role in osmoregulation, which is a process that all of us know, which occurs when solutes are accumulated in one side of two spaces separated by a selectively permeable membrane. And as a consequence of this accumulation of solutes, water tends to be intaked, to go to this compartment. And this is what happened in plants, thanks to the osmotic water potential, which eventually explained the difference between a turgid plant and a flaccid plant. Yeah, so we know that the capacity to accumulate solutes promotes the intake of water and promotes the turgence of the tissues. So, given this, we can think that to account for high levels of carbon reserves should be an adaptative trait to face drought, right? But an open question is if this is really the case. So let's take a look at the patterns. What happens with the levels of carbon reserves in trees under droughts? And let's start for this site, which is the Parque Nacional La Campana in central Chile, a picture taken in 2020 after a long and severe drought, which is still ongoing, but at that time it lasted for more than 10 years. And after these 10 years, we started to see all this evergreen forest in the color that you can see now in the image. Yeah. So ecologists started to be really worried, and we initiated a study there with my colleague Susana Paula, where this is a very diverse forest, a Mediterranean forest, where we sorted the species according to the degree of decay. Sorry that this is in Spanish, but here is the defoliation and the loss of greenness, which together were considered to create an index of decay. And we looked at the carbon reserves levels of the branches of all these species, comparing trees that within each species were in a kind of control conditions, or not that bad condition, and a very, very affected condition. Yeah, so we have the levels of non-structural carbohydrates, starch, and sugars, in the two status, in species here that were really severely affected by the drought, in trees that were not that bad, and within the same species, in trees that were much more visually affected. And then we have the same concentrations for the species that were located here, species that were apparently dealing with drought in a much more successfully way. And what we found is that when we compare the condition of stressed or more affected trees, those of showing severe decay were really had significantly lower concentrations of non-structural carbohydrate and sugars than drought trees of the sorry, this is, oh, sorry, this is, I'm very sorry, this is actually the opposite, right? So this is, switch this in the x-axis, in the here. So these are the less severe, yeah, and these are the more affected trees. So we know that the less affected species, and especially the more affected trees of the less affected species, had a higher carbon reserves and sugars than those species that had a higher decay. So very sorry for this. Please consider to switch this. I will switch it if possible in the presentation that you will upload. So and then we have a similar pattern where, so here we have, of course, more carbon reserves in the trees that are in a better conditions, yeah. And a similar pattern was recently showed by Sinery Muller in a paper of Nature Communications, where they compare the carbon status of trees in Brazil in sites of contrasting precipitation. And they found that in those sites, in the drier sites, we are like Fek, Ken, Man, that you can see here, that are those sites that experience much lower precipitation in summer. The proportion of sugars represented in the total non-structural carbohydrates was significantly higher, and also the condition of drought experienced, of course, by these trees, when compared to those of the more humid sites. And with the starch happened exactly the opposite. So again, this is evidence showing that, and something important that I have to say, this is a study that shows this increase in the proportion of sugars for the dry season. When the comparison between sites is made in the wet season, they found no pattern. All the trees have the same level of sugars and non-structural carbohydrates. However, in the dry season, there is a capacity of these trees, or there is, we don't know, the requirement to increase the proportion of sugars. So far we can think that, yeah, to have more sugars is definitely advantageous to face drought and could be potentially adaptative. However, we know that at moderate drought, trees often have more carbon reserves, and this is because growth is more sensitive to drought than photosynthesis. So when we increase the level of drought stress, for example, we decrease the water potential, we can see that both growth and photosynthesis will decrease in the different species. However, in all cases analyzed so far, the decrease in growth occurs much earlier than the decrease in photosynthesis. And therefore, high level of carbon reserves under moderate drought could just reflect this plastic response to a carbon surplus. Yeah. So an open question is whether high carbon reserves increase under drought is a plastic or a genotypic adaptative response. If it is a plastic, we could expect that it is expressed only under drought, and we should find no difference between genotypes from different sites when we compare them under adequate watering. By contrast, if the trait is adaptative, we should expect that genotypes from dry and humid environments will differ in the level of carbon reserves. Yeah, those from dry environments should exhibit higher carbon reserves than genotypes from humid environments when we compare them in a common garden, which is what I did. But before that, I wanted to show you that we also demonstrated in 2017 that sugar accumulation is indeed an acclimation response to drought. And we demonstrated this using pine plantations. So what's the advantage of pine plantations? Well, that the pine plantations used in Patagonia were planted along this impressive precipitation gradient, and they were also coming from a same genetic source. Therefore, we compared Pinus ponderosa and Pinus contorta at two contrasting climates, a mesic and a dry site. You can see here the water balance at these two sites. I am there sampling roots, and here is Alex Fajardo, my colleague, also coring the trees. And here is Gunther Hasch, a colleague from Basel University who came to Chile and participated from this study and is sampling the branches. So we found here that the growth of the trees was, of course, low in the dry locations for both species. But in any case, except needles, but for the branches, stems, and roots, we found that trees were indeed having a carbon surplus, so supporting the idea that this is, at least we know from this study that this is a plastic response. And we know this because the genetic origin of the pine was the same. They were not drought adapted and humid adapted populations. They were coming all from the United States, from the same nursery. So we do know that they are a plastic response, the accumulation of carbon reserves, but still the question of if high NSC and SS reflect local adaptation to drought is open. And another open question is whether higher concentrations of non-structural carbohydrates come at the cost of lower growth, which is something that has been recently proposed, given that the same amount of carbon cannot allocate it to similarly at the same rate to different functions. So to test this, or to address this question, we took advantage of the exceptionally wide niche breed tree species, Embotrium coccineum, which is a treelet that you can see here in this picture, which naturally is distributed in the very humid parts of Patagonia, with a precipitation greater than 3,000 millimeters per year, in more mesic site, and in the limit with Argentina, where precipitation is lower than 500 millimeters per year. We established a common garden experiment after seed collections from the two contrasting precipitation sites, so from the dry location and from the humid location. We skipped this location for the common garden experiment. And after 3 years, we started to harvest the seedlings in spring, in summer, and in autumn to measure non-structural carbohydrates, starch, and soluble sugars concentrations in the different tissues. We also measure biomass, and we apply linear mixed model testing for the effects of the climate, of the origin of the population, and the season. Yeah, this is the date of harvest. And we consider the seed site as a covariate in all the models. We found first that plants from the wet location were significantly bigger than those from the dry location. And then we generally found that the carbohydrate concentration, you can see here non-structural carbohydrates, starch, and sugars in the leaf, stem, and roots, and for the 3 seasons, was overall for most, for almost all the cases, all the combinations of tissues and season, not different between the dry populations and the moist population. So please put your attention just in the size of the 2 bars for each date. Yeah, and you can see that generally they don't differ. Yeah. However, we found 2. So first, I would like to say that here we found that in general, in springtime, concentrations of all the compounds were higher than for the other seasons. And this is because in springtime, evergreen species can take advantage of the warmer days and sunny days before they start growing. Yeah, so this is just a consequence of a favorable environmental conditions for photosynthesis before the growth has been significantly reactivated. Yeah. Then we see this very significant decrease in all the tissues and for most of the compounds in summer and in autumn, which reflects that the carbon balance become more negative. And this is because plants started to grow. So they started to really use their reserves. But what I wanted to emphasize in this picture is here you can see that there is a significant difference in the response of each genotype to the season. Yeah, so even when both populations increase the sugars of the roots from spring to autumn, we see that this increase is steeper in the populations from the dry locations. And as a result, in autumn, for example, we have significantly higher root sugar concentrations in the dry than in the humid genotype. So the model supported this pattern, or actually demonstrated this pattern with a statistical significant interaction between the date and the origin of the plants. This is the population. And the other significant result, was that even when both populations decreased the concentration of starch in the roots during the season, the humid population kept significantly higher concentrations than the dry population. So let's now move to the final part of the presentation and discuss this result. In first place, we can answer the question about if do high NSC and SS concentration reflect local adaptation to drought? No, because we didn't find a significant difference between the two populations. And actually, as I previously showed, we found that starch was greater in the humid population, contrary to what is expected from local adaptation to drought. The second question is, high concentration come at the cost of lower growth? Of course, no, because the previous answer was showing no difference in concentration between the two populations. So by default, our results suggest that higher NSC concentration in plants at dry climates, as has been reported in several studies, are reflecting carbon surplus driven by the higher sensitivity of drought than photosynthesis to drought. Therefore, we can indicate that there is no role of drought as a selected driver of a growth storage trade-off. Thus, at least in this species, it doesn't seem to be a competition between growth and storage for carbon. Or put in other words, it seems that the carbon availability is sufficient to meet the demands from growth and from the formation of carbon reserves. And this is important because then we can anticipate that growth stimulation under increasing atmospheric CO2 concentrations in trees of arid environments should therefore not be expected. Then we also can say that seedlings population from contrasting climates did differ nonetheless in the seasonal dynamics of sugars concentrations in roots, and this is consistent with local adaptation to drought. We say this because increasing concentration from summer to autumn, as we found in this case, could be locally advantaged to tolerate dehydration during late summer. It is important to remind that precipitation, the lowest level of precipitation in all Patagonia across sites happen in late summer. And we therefore suggest that increasing concentrations of sugars at the driest period reported in other studies, like the Sinari-Muller and the Paula and Piper study that I showed at the beginning of the presentation, could therefore be And also, if we saw results from the study of the meta-analysis of Martinez-Vilalta that I showed before, we can see that Mediterranean plants are the only plants on Earth that present a consistent increase of sugars represented in the green color during the summer, which is the driest period in Mediterranean biomes. Therefore, our study leaves open the door to this possibility. Could be an adaptative response to drought. And finally, I will finish the presentation saying that higher sugar requirements of the dry ecotypes were not met from higher photosynthesis, where at least unlikely, this is unlikely because even when we didn't measure photosynthesis, we measure the leaf nitrogen concentration, which is a very precise surrogate of the photosynthetic capacity. And we found that the concentration was the same between the two populations. So this means that increases in sugar in dry ecotypes comes from most likely from some starch degradation, which may limit the capacity of dry ecotypes to respond to other kind of natural disturbance like herbivory, which is a very, very important stressor in the humid, in the temperate rainforest, where the humid ecotype thrives. And therefore, we can expect that the dry ecotypes having less starch, probably because some of that starch is used in the formation of sugars, are less adapted to disturbances like herbivory, less tolerant. So with this, I finish. I would like to acknowledge to all the people that in some or in other way has made a very, very important contribution in fieldwork, in data analysis, in administrative parts. And I will also like to mention my collaboration with Professor Ana Sala from the University of Montana at Missoula, who is always very willing to have very fruitful discussions with me about this topic. And with this, I finish. I would also acknowledge the funding from these different projects mentioned or illustrated here. And thank you for your attention and for your patience, because I took more time to present.
That's good. Thank you very much for your talk. It was a nice story that a nice wrap up at the end where we could see the whole story and the link to herbivory. I think a lot of the students in the class will enjoy perhaps talking about that. Are there any questions from the audience? I can go while people prepare their questions. So some of the work go ahead, Sylvain. I'll ask my question if I have time later.
Thank you, Audrey. Hi, Frida. Hope you.
Hi, Sylvain.
How are you doing?
Yeah, I'm fine. Nice to see you.
I was wondering, you never went there, but when you explained that, and I totally agree that photosynthesis is affected before, growth is affected before photosynthesis, and you have this accumulation of carbon, you never mentioned that what is done with this carbon after. I mean, is it adaptative still, that accumulating this carbon could be used for regrowth and recuperation, recovery of growth after that? And I really want to have your opinion on that. And if we look at long-term growth, how those period of carbon accumulation could recover and have better growth, and finally get similar seasonal growth if the tree is using it. But what is your feeling about that? Those trees are using this accumulation of carbon, or they do that and finally they are lost?
Yeah, well, yeah, thank you for those questions. They are really for me, these are ongoing questions or ongoing answers. Yeah, I'm still building the picture. Yeah, I do think that carbon reserves are passively accumulated, or they represent a carbon surplus when you have or after you have a threshold of carbon in the atmosphere or within the tree. However, under carbon stress conditions like deep shade or like very, very reduced CO2 atmospheric concentrations, which can be manipulated through experiments, there is evidence that plants priorities the allocation to storage. Yeah, so I think that distinction is important. Yeah, one thing is when we are trying to predict carbon sequestration by trees, big tree, adult trees in most natural conditions, yeah, where usually the main limitations are droughts and temperatures, yeah, except for seedlings that also face light limitations. But when we think in temperature and drought, I think that most of the dynamics of seasonal accumulation that we observe represent accumulation, passive accumulation. Yeah, but this is like a fraction. Yeah, like the upper fraction of the cake. Yeah, if we see the dynamics when there are carbon limitations, for example, driven by light, seedlings in low light, or trees that were recurrently defoliated, but then have the chance to recover photoassimilates because the disturbance goes away. I definitely think that carbon reserves take priority over growth. Yeah, but for the specific case of droughts, I think that I think that the seasonal dynamics respond to passive accumulation, the allocation. However, I do think that there is, like I show in this study, a genetically mediated or adaptative regulation on the fractions. Yeah, on the fraction. It's obvious, even from the plastic response and from the my own study and these other studies, more global, or the Brazilian study, it is obvious that even under drought, trees are definitely choosing to store sugars, and this is not passive. There is a strong regulation behind that response, and I'm showing here that regulation is probably genetically driven, not only an acclimation response, but is constitutive. So to finish, the response of growth to carbon, to CO2 atmospheric concentrations, that's probably more complicated, because what I have seen is that many trees prefer to feed their growth from current photosynthesis, not from storage. Yeah, but that doesn't mean that storage is in purpose kept. Yeah, we don't know that.
Thank you. Yeah, it's an ongoing question, and I saw on several species here that after growth, some trees that have experienced a drought have a higher growth than the standard. So they are using this carbon, but sometimes we also find that they are dying full of carbon. So it's like they don't use it. They have this period they could accumulate, and they could store it for a better moment. But when the better moment comes, they don't use it and they die. So yeah, that's still something to explain. But thanks to your work, I mean, I'm using more and more of your work in my courses. It's amazing what you are doing with Alex, and I'm following you like a fan.
Oh, thank you. Thank you for your, how do you say?
You can say kindness, maybe.
Yeah. Yeah.
Are there any more questions from the students in the class? We can follow. You can keep your questions, and we can ask them at 2:00. Anything else? I have a few, but I'll keep them for the discussion period as well. Okay, so thank you, Frida. It was so much material. I think it's a lot of material to digest, especially when it's not exactly our field, but very interesting. Yeah, and a very nice story that you told. So thank you for that. And thank you for everyone being online.